Hi everyone. This will be the last urinary system physiology or renal physiology lecture. Before I get started, I wanted to give you some kudos for studying and learning the material under these circumstances. You guys did rather well on your tests. And I know that it is impossible to do well on those tests without having studied. So you simply wouldn't have had time to finish the material if you hadn't studied it. So good job. I realize these aren't ideal circumstances, but I wanted to give you some props for choosing to work. I am on your team. I will help you get through this. We've got just one lecture test left to do, and then a final. The final will be comprehensive. I am working on getting your prior tests up so that you can review them. I will get all of the test questions up there. I probably won't be able to get you your answer sheet. I just don't know how to do that for test 2. But I will get the questions and the answers so that you can actually look through those, because a lot of the final exam questions come from old tests, at least 60% or so. So expect an update on that within the next little while. I've got to get this stuff done first, but okay. And then do remember that my unofficial official policy is that by the time the final comes, if you are making an A average, including all of your tests without dropping one, it means you have to have taken the 4th test by then, your quiz scores, and then if all of those average out to a 92.5 or higher, then you can be done. So that's some good motivation for some of you guys who are kind of at that point, to just keep going through the end of the semester. I think you really will have gotten the vast majority of what you would have gotten in an in-class physiology, except fewer jokes and fewer stories because there's nobody to really respond. So sorry about that. Some of you guys were tired of my jokes anyway. Okay, so let me pick up something that I forgot the last time and remind you sort of big picture of where we were. So what we were talking about last time was we said that basically excretion, go back to this bad picture, is what you end up eliminating via your urine. And excretion is filtration, pressure driven, we talked about that, minus reabsorption plus secretion. And so what we are still working on is reabsorption. Last time I did sodium reabsorption and potassium reabsorption and some of the negative ion reabsorptions. I accidentally skipped the nutrients like glucose, so I'll show you that one now. And then we're going to have to do water reabsorption. That one is probably the most challenging, but if you just take it slow, you will get that one as well. So remember that as far as the locations we were looking at, oops, not that one. That one's good too, but we basically did filtration here. And just a reminder, filtration moves things, oops, lost my picture there, moves things from wet fluid to wet fluid. Filtration moves things from the blood, which is higher pressure, to the filtrate, which is lower pressure, or from the capillary tube to the P tube, the uriniferous tubule. And specifically, it's moving it into this portion right here called the glomerular capsule or the Bowman's capsule. Okay, filtration is pressure driven. It's not selective at all except by size. And then what we were talking about next is we were talking about reabsorption. And reabsorption is going to move stuff the opposite direction, right? It's going to move things from the tube, the uriniferous tubule, color-coded P color here, back into the bloodstream. And that's what reabsorption is. And reabsorption, we said last time, was basically moving things by whatever mechanism you could move them, the cheapest, fastest way. If you can move them by diffusion, do that. Most things don't move by diffusion, but some things do. But if you needed it, you would use a channel, or then a carrier, then active transport, secondary if you could do it, primary if you couldn't. And then I told you that I still didn't find any evidence that endo or exocytosis is used at the kidney. And so what we were basically talking about is we were getting some examples of stuff that we learned all the way back at the beginning of the semester, in which you're doing epithelial transport, in which you move something from the lumen and then you move it across the apical surface, across the intracellular fluid, across the basal surface, into the extracellular fluid. They call it peritubular fluid, but it's just interstitial fluid. And then across these fenestrated capillaries into the blood plasma. And the mechanism that you use here is based on the chemistry and concentration of the solute that you're trying to move. So now what I want to talk about moving is I want to talk about moving glucose. So glucose does get filtered out at the glomerulus. It definitely does. It is water soluble, and it's not huge, right? It's not tiny like an ion, but it's not big like a protein either. So it gets pushed through the holes, the fenestrated capillaries and those filtration slits, and it moves into the tube. And how much glucose would we like to retain, if we could, back into the bloodstream? How much glucose do you think you'd like to pull back from the tube into the bloodstream? All of it, if you could, right? So glucose is basically the equivalent of money, right? Remember, I called it the hottest of all the molecules. So what we are going to do is we're going to try to reabsorb as much glucose as we possibly can. Under normal circumstances, 100% of your glucose is reabsorbed. Now, that might not be true if you go on a bender with Halloween candy, or of course if you are a type 1 diabetic, but let's talk about how you would move glucose back in. So again, our objective is to move glucose from the tube back into the bloodstream, right? It's glucose reabsorption. So let's see how that is going to occur. So it should be super duper familiar to you, because glucose reabsorption is trying to get everything to move, this direction. So this is your net direction that you're trying to head. And how are you going to do it? Well, you're going to do it the cheapest, fastest way that you can. And what we're going to do is we're going to do secondary active transport of glucose. And then we are going to move it across the basal surface by a different mechanism. So here's glucose, right? In the tubular fluid, or the filtrate, right? And we pushed it out, and now I need to bring it back in. And we need to bring it back in regardless of glucose's concentration inside the cell. We still want to bring it in, even if there's a lot of glucose inside the cell, because we just brought it in, excuse me. So what we are going to do is we are going to allow glucose to hitch a ride with something that is predisposed to going inside the cell because of its electrochemical gradient, and that's our friend sodium, right? Remember how hard you work to keep sodium concentration low inside the cell, and although it's not shown here, you guys remember that the outside of the cell has a slight positive charge and the inside has a slight negative charge. So that's the motivation for sodium to go in. So what we're going to do is we're going to move sodium, but instead of moving it through a channel, which it normally would, now I'm going to move it through a carrier, and I'm going to let it bring a hitchhiker, the hottest hitchhiker, glucose, with it. So I'm going to bring it in, and then how do I get glucose back out? So let's follow glucose for just a second. Following glucose, how do I get it back out of the cell so that I can get it through those fenestrations in the capillary into the bloodstream? Well, this was a carrier, right? And this is a carrier. Does anybody remember why glucose needs a carrier instead of simple diffusion straight through, or instead of diffusion through a channel? It needs a carrier because it's mid-sized and water soluble. So now this carrier, it just moves it down its concentration gradient, because glucose is going to be high here because I just brought it in. I never let it stay high here because it's got work to do, right? So I carry it away. So high here, low here, move it through a carrier, and then let it diffuse straight through the fenestrations. But of course, the other thing is that, remember, we have to fix the screwed up sodium concentration gradient that we just created. So we are going to do that via the sodium-potassium pump. So this is basically just taking secondary active transport of glucose that you learned all the way at the beginning of the semester and putting it into the context of glucose reabsorption. And glucose reabsorption happens all throughout the tube, primarily at the proximal convoluted tubule, but in its secondary active transport. But it is subject to saturation because this is a carrier. So what is going to happen if you have so much glucose in your urine because you ate 6 Butterfingers right after Halloween, or you are a type 1 diabetic who has really high sugar? What's going to happen to your glucose? Can you bring it all back in if your glucose carriers are saturated? No, they can actually get full. And if they get full, let's go back to this figure for just a second. If they get full, what will happen is, so I'm going to, again, filter glucose out. I'm going to pull the vast majority of it, hopefully close to 100%, but maybe I can't because I ate 6 Butterfingers. I'm going to pull the vast majority of it back into the bloodstream at the proximal convoluted tubule. But let's say I ate 6 Butterfingers or 16 Butterfingers, right? Then I couldn't bring it all back in, and now I am delivering to the loop and then to the distal convoluted tubule a relatively high glucose concentration filtrate. And what that is going to do is you're going to end up with glucose in your urine, and that is called glycosuria right here. And since the glucose is going through the tube, it is exerting an osmotic pressure on the fluid around the tube as it goes through. And what it's going to do is it's going to pull water into the tube, into the uriniferous tubule. And then what's going to happen is you are not only going to have glycosuria, you're also going to have polyuria, which is why, for instance, a type 1 diabetic who had really high sugar, or if they hadn't been diagnosed yet, one of the first indicators is that you start peeing like a racehorse. And why? Because all of the glucose that you're not taking into your cells, you try to reabsorb it, you fail at reabsorbing all of it, and then it goes through the tube and pulls water into the tube, and that water in the tube ends up getting peed out. So that's glycosuria and polyuria, both associated with high sugar in the bloodstream to the point that you can't do reabsorption completely. Now, that's not the only nutrient, by the way, but of course, the other nutrients, amino acids, sometimes lactic acid, some water-soluble vitamin, which isn't a nutrient, but same idea. A lot of those are reabsorbed by the same mechanism, which is secondary active transport. Okay, so that is glucose reabsorption. All right, so now we are going to get into water reabsorption. So water reabsorption, actually there's 3 different steps of water reabsorption, so you're going to have to kind of pay attention to it. And I changed these 2 percentages because I found some better information. I won't ask you those percentages specifically. You definitely have to know the 65. So let me show you a little video to introduce you to the whole water reabsorption thing, and then I'll go through it slowly. Okay, so this is the same video I was showing last time, but it's going to start with the reabsorption and then follow into water reabsorption, and then I will go through it in the lecture notes, and then I also have a YouTube video. This guy, for some reason, explains it in a way that really clicked to me. Okay, ready? Oops.
Useful substances are removed from the tubular fluid and returned to the blood.
Sorry, let me start that one more time.
Fluid as it flows through the renal tubule. In tubular reabsorption, useful substances are removed from the tubular fluid and returned to the blood. Solutes move across the tubule wall into the interstitial fluid by processes such as diffusion, facilitated diffusion, active transport, co-transport, and osmosis. The solutes readily enter the surrounding capillaries to return to the general circulation. Water follows the solutes by osmosis.
Okay, so
65.
First big thing is that I spent a lot of time and money moving solutes, moving sodium, moving potassium, moving glucose, moving all of those. And by doing that, I create an osmotic gradient for water to move back in. But it's not as simple as that, so let's watch.
10% of the water is reabsorbed from the proximal convoluted tubule. An additional 15% from the descending portion of the nephron loop, and an additional 19% from the distal convoluted tubule and collecting ducts. Thus, 99% of the glomerular filtrate ultimately returns to the bloodstream. The 1% or less that is not reabsorbed will be excreted as urine.
Okay, so this is water, okay? Look at the proximal convoluted tubule. 65% of the water that you push out via filtration is reabsorbed at the PCT, and that's osmosis. And then you are going to have the capacity to reabsorb up to 15% of it at the loop. And then this last little bit is the 19% that you can get at the distal convoluted tubule and the collecting duct. And this is the one that is regulated primarily by ADH. So we'll get to that in just a second.
In tubular
okay, we'll come to secretion in just a minute. All right, so basically what we are going to have is 3 steps of water reabsorption. Proximal convoluted tubule is going to be the first step. You have no control over that. What happens is at the proximal convoluted tubule, as you reabsorb all of those ions, especially sodium, water is going to passively follow because you are creating a concentration gradient for water as you move sodium. So the PCT reabsorption is 65% of the sodium was reabsorbed in the PCT, so not surprisingly, 65% of the water actually follows that. This is non-regulated reabsorption, meaning I can't do anything about it. I always push it out and reabsorb 65% of the water at the proximal convoluted tubule. And there isn't any way hormonally with your intrinsic mechanisms to regulate that. But then what we are going to do is we're going to do loop of Henle reabsorption. And the loop of Henle is primarily just about reabsorbing water. And it's a little confusing. You may have to watch it more than once. It's actually kind of cool, but it is a little confusing. So what is going to happen, let's see if I, oh yeah, they have a picture in your textbook. Pull up this picture in your textbook. Okay, so I'm just going to give you a preview of things to come, and then the video will actually really show you. I'm not going to hold you responsible for the actual amounts, but here's what's going to happen. As you go from the proximal convoluted tubule and then you go descending and ascending loop, this is drawn a different way than the other one, but we'll be okay. So proximal convoluted tubule, descending loop, ascending loop, here's what's going to happen. On the ascending loop, you are going to actively pump out a whole bunch of ions from the tube into the interstitial fluid, into the extracellular fluid. So you're going to be doing that in the ascending loop. And what that's going to do is it's going to create a really salty situation out here, and the descending loop is really permeable to water. So what happens is I pump a whole bunch of ions out, and then I create this really salty situation, and then water gets sucked out of the tube. All right, so let me show you. So the ascending loop actively pumps solutes, sodium primarily, but others as well, into the ECF. And the descending loop is only permeable to water. It's not permeable to solutes. So we create a really salty situation. And since they're running at opposite one another, that counter current exchange makes it even more effective. So let's watch this. Ready? Okay.
The countercurrent multiplier, also known as the countercurrent exchange system, is the method that the nephron uses to concentrate urine. For a lot of people, just the name of this process is daunting. Countercurrent multiplier sounds like a complicated math formula or something, but it's really much easier than it sounds. To begin with, let's look at the anatomy of the nephron and its place in the kidney. If you recall, the kidney is divided into the outer portion, the cortex, and the inner portion, the medulla. The glomerulus and proximal tubule are located in the outer cortex. The loop of Henle descends down into the medulla of the kidney, loops around, and comes back up. By the time it becomes the distal tubule, it's back in the cortex. The collecting ducts then descend back down into the medulla and eventually empty the filtrate into the calyces and renal pelvis. For starters, I want you to think of the medulla as being very salty. In fact, at the bottom of the loop of Henle, the sodium concentration is about 300 milliequivalents per liter, which is double that of the blood. The loop of Henle has some special characteristics that explain why that is, and it's really all about saving energy. So the descending loop of Henle is extremely permeable to water, but it's impermeable to ions. So water is able to passively diffuse out of the descending limb, where it is reabsorbed by the vasa recta, the vessels that surround the loop of Henle. The ascending limb, especially the thick segment, however, is the opposite. It's impermeable to water, but will use energy to actively pump ions like sodium and chloride out of the tubule into the interstitium of the medulla. When it does this, it creates a hypertonic environment around it. Because the descending limb is very permeable to water, water leaves the descending tubule without expending any energy because it is following the concentration gradient. So it is called countercurrent because the filtrate flows in opposite directions, down the descending loop and back up the ascending loop. It is called a multiplier because the active pumping of ions out of the ascending tubule multiplies the amount of water that is reabsorbed from the descending tubule. As more water is pumped out, the filtrate becomes more and more concentrated.
Okay, that is linked in your notes if you want to watch it again. I think it really explains it very well, but I'll go ahead and go through one more time. Okay, so basically what we are going to have is, let's look at this figure for just a second. Okay, so let's look at it in the context of this nephron. So what we have here, this is the proximal convoluted tubule, this is the descending limb, and this is the ascending limb, okay, of the loop or loop of Henle. So what we are going to see is that the ascending limb right here is going to be pumping, let's write it down. It's going to be pumping ions out, okay? That one is going to be pumping ions out, okay? And then what is going to happen, and this is not permeable to water, this one is permeable to water, and it's going to create a really, really salty situation here, so that what happens at the descending, did I say? Yeah, descending, the ascending pumps ions out and the descending is permeable to water, and you create this really, really salty situation. So that in the descending limb right here, water moves out. Okay, so let's show you. Okay, so what we end up doing here is we actively work on pumping ions out, and then we passively get water out. And the longer the countercurrent, the loop, like in desert animals, like I mentioned before, the more effective you are at water reabsorption. So this is the countercurrent multiplier system. And what it's doing is it's concentrating the urine by actually pulling water out of the urine. Okay, so now we are to our third time that we are going to work on water reabsorption. What we did here is osmosis just passively moved 65% of the water that was filtered out back into the bloodstream. And then here we actively work on pumping ions out in the loop. And then we get passive water reabsorption here. And then this guy right here, the distal convoluted tubule, you can regulate or select how much of the water that's left over, which is up to 19%, how much of the water that's left over you want to reabsorb. And so we'll talk about that. So at the distal convoluted tubule, and also the collecting duct, but I'm just going to say DCT. At the DCT, what we are going to be able to do is regulated reabsorption of water. None of these are regulated by hormones that we just talked about, but this one is regulated by hormones. This occurs in the cells of the distal convoluted tubule and the collecting duct, but only if ADH is present. So ADH was released from where? Who guys remember? Who remembers? ADH comes from the posterior pituitary, and you released ADH from the posterior pituitary, and you always knew that it targeted the kidney, but now I'm specifically telling you that it targets the DCT and collecting duct of the kidney, DCT primarily. So what's going to happen is if you don't have ADH in the distal convoluted tubule, even if sodium ion was happening, a reabsorption was happening in the DCT, water can't follow it because this thing has tight junctions. And in order for water to follow sodium and solutes in the distal convoluted tubule, you actually have to have ADH. So water can't pass through the DCT cell membrane unless you have ADH, even if sodium is going through the DCT cell membrane. So you guys remember, I hope, that aldosterone was the hormone that will cause sodium reabsorption at the distal convoluted tubule. But now what I'm telling you is if ADH is not present, water can't follow sodium. So if ADH secretion was low, then you would not have much water reabsorption and you would get polyuria again. This time the polyuria is completely unrelated to glucose concentrations, but you are going to pee a lot, and that of course is diabetes insipidus, right? When you have chronically low levels of ADH, so you can't do that last 19% of water reabsorption at the distal convoluted tubule, and you will pee like a racehorse then too, only it won't have a lot of sugar in it. So there is a text box in your textbook about diabetes insipidus. I would read it if I were you. Okay, but what if I was secreting a whole bunch of ADH? So I'm secreting anti-diuretic hormone because, I don't know, I am chronically dehydrated, like I totally am because I'm drinking coffee instead of water right now. Okay, so what happens if ADH secretion is high? Then what happens is it gives permission for the water to follow the sodium at the distal convoluted tubule. So as you reabsorb sodium at the distal convoluted tubule, if ADH is present, water will follow, okay? So water reabsorption is going to be high, your final urine volume is going to be low, and your urine is going to be really, really concentrated, and ADH will also cause a thirst response. So one more time, at the distal convoluted tubule, if you don't have ADH, you will not do that 19% water reabsorption. If you do have ADH, you will do up to that 19% water reabsorption. Okay, so, come on. So what controls ADH release? This is all the way back to the endocrine chapter. We can reinforce this. ADH release is primarily controlled by the water concentration or osmolarity of the blood as it hits the hypothalamus. If the water concentration of the blood was low, like the blood was slightly hypertonic, then the posterior pituitary actually releases ADH and then the water is reabsorbed. But if the water concentration is high, because you're one of those people who drinks a lot of water, mad respect for you, if the water concentration is high, then it will inhibit ADH release, and therefore you will let that 19% go at the distal convoluted tubule and the collecting duct. Okay? All right. One last time. I know you guys are tired of talking about water reabsorption. 3 steps in water reabsorption. 65% here happens by osmosis. You can't do a damn thing about it. This portion right here, the countercurrent multiplier system, happens in the loop. That's up to 15% more with a longer loop, and that's primarily because of active pumping of solutes into the extracellular fluid and water passively following via osmosis. And then at the distal convoluted tubule, you've got one last shot of water reabsorption, but only if ADH is present. Okay, so now let's look at reabsorption of other substances. So whatever I pushed out via filtration, I have potential to reabsorb it. And so we have talked about sodium reabsorption in detail, right? That's primarily controlled at the distal convoluted tubule by ANP and aldosterone. At the proximal convoluted tubule, it's obligatory. You can't do anything about it. Then we talked about glucose reabsorption as much as you can by secondary active transport. Potassium reabsorption, you always lose 10% or more of your potassium. And water reabsorption, we just talked about in a lot of detail. So what about other stuff? So if it's a lipid-soluble something and you push it out by filtration, it will move back in if it wants to, down its own concentration gradient. So lipid-soluble substances are reabsorbed really easily. Reabsorption is not regulated. You have very little control over it. So if you push a fatty acid out and it wants to diffuse back in, it sure as hell will. Lipid-soluble drugs, lipid-soluble toxins. For that reason, your kidneys kind of suck at clearing lipid-soluble poisons, toxins, whatever out of your body. The liver can help because if it's lipid-soluble, the liver has the capacity for some things to change it so that it's more water-soluble, less lipid-soluble. If it can do that and then it can send it to your kidneys, then the liver and the kidneys can like be a team for getting rid of lipid-soluble stuff. But what if a substance is not lipid soluble, doesn't have a carrier molecule, too big to be reabsorbed? Then when you push it out, it usually stays there because there's no mechanism to bring it back in. So urea, uric acid, if you push them out, they either stay out there completely or there are some reasons why you would reabsorb a little bit of urea. Creatinine from creatine phosphate metabolism has no reabsorption mechanism. So generally speaking, I mean, there are exceptions to this, and you can learn it in more detail a little later, but not in this class. But if you push it out and there's no mechanism for reabsorption, then it ends up being excreted. Cool? Okay, and then the last phase is tubular secretion. So we did glomerular filtration, and then we did tubular reabsorption, happens all throughout the tube, okay? And now we are going to do tubular secretion. So now we are going to be moving things again. Secretion moves things from the blood to the urine, but this is on purpose. This is selective. Like you're picking what was left in the house and what to dump out on the lawn. You're getting rid of waste. You're getting rid of an extra 70 cans of corn or whatever the hell you found in the cabinets. So you get rid of excesses and waste by this mechanism. And so the secretion, whereas reabsorption, so filtration occurred here, reabsorption occurred all over really. Secretion is primarily, there are exceptions, but primarily the distal convoluted tubule, and then some of it is in the collecting duct. But it kind of depends on what you're secreting. So the two major effects of doing tubular secretion is to get rid of waste products that you didn't get rid of before, like the ammonium ion. There are some breakdown products from medications that you get rid of by secretion, penicillin, phenobarbital. Urea and uric acid, if you reabsorbed it or you didn't get rid of it completely, you can get rid of it that way. But then we also do potassium secretion, and potassium, remember that when I use aldosterone, I reabsorb sodium, but I secrete potassium. So if I had excess potassium, like I was hyperkalemic, I would get rid of it if aldosterone was present. And you guys remember where aldosterone comes from? It comes from blank. Where does it come from? It comes from the adrenal cortex. And of course, it's exchanged for sodium. And then I can also decide to secrete H plus or not secrete H plus, depending on the pH. So you can control blood pH by actively secreting H plus, either by itself or exchanging it for sodium, because this process that you've seen before, this freaking equation that's been with you all damn semester, right here, this little bugger, this occurs in your kidney tubules. So inside the epithelial cells of your kidney tubules, what you do is you force carbon dioxide into H plus and HCO3 minus, and then you decide at the kidney tubules whether you want to hold on to this or get rid of it, and whether you want to hold on to that or get rid of it. And by doing that, you can have the kidneys have a major impact on the pH of the body. So if, for instance, the blood was too acidic, you will actively secrete the H plus, okay? If the blood was too acidic, you would actively secrete the H plus and reabsorb the bicarbonate ion back into the bloodstream so it could help with buffering other things. But if the opposite was true, you might actively reabsorb the H plus. Reabsorb the H plus so that you could use it to manage blood that was slightly alkaline. So basically what's going to happen is the kidneys are going to look at the blood pH to decide, after they force this equation, to decide what to do with the components of the equation. And so your kidneys work hand in hand with your lungs in controlling body pH. So let's watch the rest of this little video, and then I will summarize filtration and secretion for you.
Reabsorbed will be excreted as urine. In tubular secretion, substances move from the capillary blood to the tubular fluid. Waste materials, such as metabolic byproducts, excess ions, and drugs, diffuse out of the blood to the interstitial fluid and are then transported across the tubular wall and secreted into the tubular fluid for excretion. Tubular fluid that enters collecting ducts is called urine. Depending on the need to eliminate or conserve water, collecting ducts reabsorb water to produce dilute or concentrated urine. For example, when water intake is high, the kidneys produce a large volume of dilute urine. Conversely, when water intake is limited, producing a large volume of dilute urine would lead to dehydration. In this case, the kidneys conserve water by producing a small volume of concentrated urine. The kidney's ability to concentrate urine depends primarily on two key factors. One, maintaining a high concentration of the solutes sodium chloride and urea in the renal medulla, which is accomplished by the countercurrent functions of the nephron loops. And two, the presence of antidiuretic hormone, ADH, which makes collecting ducts more permeable to water.
Okay, so that was kind of an overview of all of it. So I want to summarize this for you to talk about what is reabsorbed and secreted, just big picture, putting it all together. What is reabsorbed in the proximal convoluted tubule is pretty much whatever's going to be reabsorbed in the proximal convoluted tubule, because you don't have any control over it. So usually 65% of water, usually 65% of sodium, and usually 90% of potassium, and as much glucose as you can get, right? So that's just kind of what happens mostly here. There are other regions, but mostly here. So basically, you really don't have any control over what happens at the PCT, no hormonal control at all. What you do have is potential for hormonal control of what happens at the DCT and the collecting duct. So let's summarize what happens in the DCT and the collecting duct. In the distal convoluted tubule, if aldosterone and ADH are present, you are going to move all of this stuff. So, of course, sodium will be reabsorbed in the DCT, from the DCT back into the bloodstream, and then into the blood. And then if aldosterone is present. If ADH is present, water will follow sodium, okay? So, and then what's going to happen with all of this is that anytime you move sodium and a lot of positive ions, you'll move chloride and bicarbonate, because you create an electrical gradient for those moving. So the negative ones follow the positive ones all throughout the tube when the positive ones move. Or you can balance out the movement of positive ones by flipping them for another positive one. If you reabsorb sodium with aldosterone, you can dump potassium or dump H+. Okay, so the summary of the summary is this. So here's to summarize. Whatever happens at the proximal convoluted tubule happens at the proximal convoluted tubule. But in the distal convoluted tubule, when you have ADH and aldosterone, then what are you going to do? You are going to reabsorb sodium, but also the negative ones, chloride, bicarbonate. You're going to reabsorb water. And then you are going to secrete potassium and H+ into the urine because of aldosterone. Okay? So that's your summary of summary. Cool. All right. So I think this is, that's the last little bit. Okay. So last little bit is how much do you pee per day and what influences how much you pee per day. So the final volume of urine generally that people, a healthy person would excrete per day would be around 1 liter to 2 liters, usually around 1,000 mils to 1,800 mils. What kinds of things impact how much you excrete, how much urine you excrete per day? Well, we're going to go back and put this together and say, okay, well, blood pressure, right? So if you have higher blood pressure, which one would it impact? Let's just put this together for me. Would it impact filtration, reabsorption, or secretion? So high blood pressure impacts filtration, okay, which happens here. So generally speaking, higher blood pressure, higher GFR. So higher GFR, higher filtrate, usually higher urine volume. Of course, the renin-angiotensin-aldosterone system can be triggered by low blood pressure detected in the afferent arteriole. So if your blood pressure was low in the afferent arteriole, you would secrete renin, which would interact with angiotensinogen to form angiotensin 1, which would interact with ACE from the lungs to form angiotensin 2. Angiotensin 2 impacts the kidneys because it causes aldosterone release, and aldosterone impacts what happens here. And of course, it also causes vasoconstriction, not directly related. So basically, what happens here is if you detect low pressure here, you're going to institute those mechanisms that we talked about that would bump up the pressure. So the renin-angiotensin-aldosterone system. And I put a good figure for renin-angiotensin-aldosterone system at the end of the cardio notes. If you didn't see it there, I would stick it here too, or stick it wherever it goes in these notes because we've talked about it twice. Okay, so now what else impacts your final volume of urine? The concentration of water versus solutes in your blood. So again, what we just talked about, if you drink a lot, you pee a lot, but there's a fizz mechanism in between drinking a lot and pee a lot. So if you drink an excessive amount of water, then the water concentration in your blood increases. And if the water concentration in your blood increases, what happens is it inhibits the secretion of ADH from the posterior pituitary. And therefore, the water reabsorption that could have occurred right here in the distal convoluted tubule and the collecting duct stops, and you can lose that 19%, I think, of water. So basically, if you drink a lot, you pee a lot, but if you drink a lot, you pee a lot because you inhibit ADH secretion. And ADH is not there to cause reabsorption of water at the distal convoluted tubule. But if you're dehydrated, which is the constant Melanie problem, then the water concentration of your blood is low, right? And when it hits the hypothalamus, then what happens is it's like still hypertonic, Mel, you still suck at drinking enough water. And so what happens is I will secrete ADH. ADH is going to target the kidney, but now we know it specifically targets the distal convoluted tubule of the kidney. And it says when you move sodium in at the distal convoluted tubule, water can follow at the distal convoluted tubule. So you can reabsorb that 19% potentially. So if you don't drink a lot, you don't pee a lot, but it's because of what happens here. So small amount of concentrated urine is excreted. Okay, so what else impacts how much urine you produce? Temperature does. Temperature kind of impacts everything. If you get exposed to high temperatures, like if it's 105 degrees outside, then what happens is you send more blood to the cutaneous vessels. You're going to sweat, activate the sweat response, and you're going to lose fluid in your sweat. If you lose fluid in your sweat, then you have less fluid to lose from your urine. So as the fluid in the body decreases, ADH and aldosterone are generally secreted because you lose both sodium and water when you sweat. So if you secrete ADH and aldosterone, what happens is, of course, those let you reabsorb sodium and water from the distal convoluted tubule. So if you're really hot, you sweat a lot, which causes you to release ADH and aldosterone because of the change in water and sodium composition. And then you reabsorb here. So if you sweat a lot, you don't generally pee a lot, okay? But when you're exposed to low temperatures, the sort of something kind of interesting and counterintuitive happens. What happens is when you're really cold, you do peripheral vasoconstriction, and the peripheral vasoconstriction says, hey, it's really cold outside. I definitely can't afford to lose a liver, but maybe I could afford to lose a finger. So you bring the warm blood disproportionately towards your core, keep your vital organs alive, but that increases the pressure in your core. So more blood pressure goes to the glomeruli and it increases GFR and your urine volume increases. This is why every single time you go camping and it's freaking cold outside, you definitely have to pee in the middle of the night. It's one of the most irritating things ever. Okay, and then the last little tidbit is I want to teach you how diuretics work. So how do, we'll do renal calculi too. How do diuretics work? Well, diuretics are by definition a chemical that increases the urine volume, but please remember that whatever's in your urine came from your blood pressure, blood plasma. Okay, so there are diuretics that impact PCT reabsorption. Now, I know I told you you couldn't regulate that, and you don't by any normal mechanisms, but we do have drugs that actually regulate it. And then there are diuretics that impact this one. And the ones that impact proximal convoluted tubule tend to also affect the loop. So the diuretics that inhibit non-regulated reabsorption are really, really effective because 65% of water reabsorption happened at the proximal convoluted tubule. But the weird thing is that if you inhibit sodium and water reabsorption at the proximal convoluted tubule, what you end up doing is you end up delivering more sodium and more water to the distal convoluted tubule. And then your body is going to say, hey, there's so much sodium and water here. Why don't I try to reabsorb the sodium and water from the tubule with aldosterone? And so what's going to happen is when you reabsorb sodium, so you didn't affect aldosterone at all because you did this part, but what you ended up doing is not reabsorbing very much sodium in the PCT and delivering a whole bunch of sodium to the DCT. And then your body goes, oh, I need to reabsorb sodium. Aldosterone will cause sodium reabsorption, but only at the expense of potassium or H+. So these are really, really effective diuretics, but these are the ones that you have to be careful about potassium. I don't know how to pronounce that, furosemide. This one right here, super effective at getting rid of fluid in the body, but you generally are going to have to watch your potassium composition. Sometimes they're on a potassium drip if they're on this one, because you know you are going to lose potassium. Not because it directly interacts with aldosterone, but because it delivers so much sodium to the distal convoluted tubule that aldosterone tries to do its job. Okay, now the ones that are not quite as effective, but also not quite as dangerous, are the ones that actually work on the DCT specifically. So some diuretics actually don't mess with this part at all. They mess with this part. They mess with the regulated reabsorption of sodium and therefore water in the distal convoluted tubule. And if they can block sodium ion reabsorption at the distal convoluted tubule, you can do a decent amount of that, not exactly 35% because some of it goes on here, but you can do a decent amount, not 65% though, so they're not quite as effective. So they are called potassium sparing diuretics though, because if I inhibit aldosterone at the distal convoluted tubule, then I can inhibit sodium reabsorption, but I also don't lose any excess potassium. Spironolactone is one of those. Okay, so what about some other diuretics? Alcohol is a diuretic because alcohol actually inhibits the secretion of ADH. So it's a diuretic because it's an anti-diuretic. So that's what that one does. It inhibits the secretion of ADH, which means whatever water reabsorption I could do at the distal convoluted tubule, I'm not going to do. So I can lose, what was that, 19% up there? Caffeine is a diuretic as well, but caffeine is a diuretic because it increases GFR, because it increases the blood pressure everywhere, but it's at the kidney too. And then there's one last type of diuretic called an osmotic diuretic. And an osmotic diuretic kind of works the way that glucose works in a type 1 diabetic, because if you push glucose out and you can't bring it back in as it goes through the tube, The concentration of solutes in the tube pulls water into the tube and makes you pee. But with these osmotic diuretics, what you're doing is you're taking in something that you know will be filtered out and doesn't have a reabsorption mechanism. And so it goes through the tube and it exerts osmotic pressure and you pull water into the tube. Mannitol is one of those. A lot of the over-the-counter diuretics that are sometimes given for or used for like PMS symptoms, a lot of those are osmotic diuretics. So they push it into the tube and there's no mechanism to bring it back. So that's how an osmotic diuretic works. Okay, and last little tidbit is just to talk a little bit about kidney stones. Okay, I don't talk about dialysis because I've already done it before. We did it way back at the beginning of the semester. Okay, so renal calculi or kidney stones are what happens when, go back to this figure for just a second. All right, so let me just show you this. Come here, you little booger. Okay, let me show you where this actually occurs. Okay, so you are, of course, dumping and forming urine in the minor calyces and then the major calyces. And you, what happens with renal calculi, which is kidney stones, and look at these buggers. I've got some dramatic kidney stones to show you right here. Look at them. Oh my God, those are some real kidney stones. What happens with these is if there is a high concentration of solutes, come here, stop doing that. If there is a high concentration of solutes, I don't know why it's doing that. In the major minor calyces, the renal pelvis, or the ureters, then what can happen is they can start to crystallize. Why is it doing that? Okay, there we go. They can start to crystallize. As they start to crystallize, then you get these basically rocks in portions of your kidney that then have to be delivered down your ureters. And then in your urinary bladder, a lot of times they're not necessarily going to hurt, but when they try to go out the urethra, you're definitely going to feel it again. So that's what a kidney stone is. And of course, these are generally going to have to come out of your urinary tract, and most of the time they come out by you trying to pee it out. This is the time in which women are very, very happy that they have a urethra that's only about 1.5 inches versus a male urethra that's about 8 inches long. I'm not telling you the penis is 8 inches long. From the inside, the internal urethral sphincter to the external urethral orifice is on average 8 inches in a male, 1.5 inches in a female. So there's a text box about kidney stones in your textbook. So what makes these more likely? You can have just a genetic or metabolic predisposition, and that blows because there's not a damn thing you can do about it. But there are some other things. A decrease in fluid intake. If your urine is really, really concentrated in solutes, you're more likely to form these. I'm talking to myself right now. If your urine is abnormally alkaline or acid, you're more likely to form these. If you ingest excessive amounts of mineral salts, or if your parathyroid glands, which increase your calcium concentration in your bloodstream, also phosphate concentration in your bloodstream, if those are overactive, it can cause kidney stones as well. And then there's a decent amount of anecdotal evidence, probably not experimental yet, although I haven't looked this year, that soda consumption increases the likelihood of renal calculi, especially diet soda consumption. It's even saltier than regular soda. So what can you do to reduce the likelihood of forming renal calculi? Although this seems really annoying, drink lots and lots of fluid, preferably water, Melanie, not coffee in the middle of the day, especially before bedtime. That may mean that you have to get up and pee, but it's probably better than having a kidney stone. And you can limit intake of anions like oxalate and urate, but don't limit intake of calcium and other others of your cations. Okay, so that is that for urinary system. I will post this for you. It's not supposed to be up until Monday, but I'm going to go ahead and post it early.